Okay, so I'll talk a little bit about neuroanatomy in terms of network function. So part two only refers to the fact that if you remember, I had a short introduction to neuroanatomy back in September. So this continues that. So next slide, please. So by the end of this session, you'll know what white matter tracks are and how they're measured. So there were a couple of nice videos in the learning content about how diffusion works, and it illustrates very nicely the concept. Then I have two different approaches to looking at network approach to normal brain function as well as the disease. And then through the case reports, we'll look at the relationship between these large-scale brain networks and disease itself. So next slide, please. So just to remind you a little bit from September, there's the localization approach that I introduced, and I'm sure you have seen in other talks this past 9 months or so. So one of the first studies was from Paul Broca, and he had a patient that could understand language, but I could only say the word tan. So a post-mortem examination found that this patient had a lesion in the inferior frontal gyrus. That's indicated there with the red arrow. And he made the association between the ability to speak and this particular brain region. So this region is known as Broca's area. In the next slide, please. So the next, again, another key finding from the 19th century is from Carl Wernicke. And he discovered a region in the superior temporal gyrus that would be indicated there with the red arrow. And this was associated with the understanding of language. So he had a patient that was able to speak, but it was just nonsense content. So the association was made between the ability to understand language and this particular area, which is now called Wernicke's area. So these are two key findings. There are other findings also from about this time point, presenting evidence of the localization approach. So I'll talk a little bit now about the network approach that in the last 10 years has received a lot of attention now. Earlier than in this modern, quote unquote, modern times, there have been periods when a network approach also has received a lot of attention. So if we go to the next slide. In this slide, I'm showing here in panel C. So in panel A and B, you have the two regions, Broca's region and Wernicke's region, indicated in gray. And then in panel C, in orange, you see a track or let's say a road connecting both Broca's and Wernicke's area. So this is the arcuate fasciculus. And this white matter track connects these two language regions. Now, patients may have a language impairment if there's a lesion within the arcuate fasciculus. So the two traditional or classical language regions are not impaired, but the communication or interaction between these two regions is, and this may lead also to language impairment. So this is a very nice illustration of the network effect in the brain. Okay, that interaction between regions or among a large group of regions is important for cognitive function. So next slide. So one key concept is the connectome. So instead of modeling the entire brain as individual regions, we look at the brain connectivity and as a network and try to model the network. Now, the effect of lesions is number one, both the impact it has locally in the region, but also the impact it has downstream and upstream, so to speak, to other regions that are part of the network or subnetworks. So again, it's not a localized effect, but it's the effect on the network itself. So in terms of investigating diseases or neurodegenerative diseases, looking at network approach gives us a better understanding of how the spread, where the pathology is located and how the pathology spreads through the brain, as well as how the spread of the pathology through the brain leads to the appearance of symptoms, both cognitive symptoms as well as clinical symptoms. Now, I use the word spread in terms of neurodegenerative diseases, not to mean that, that the specific pathology is moving through the brain using the white matter tracts or other connections, but it is really more an effect of one region to the other regions that are connected to it. The pathology itself, at least that we know up to now, doesn't spread by moving from one region to the other. That is in the case of prion diseases. Okay. So we're not talking about prion diseases. Okay, next slide, please. So this is, again, a very nice illustration of the white matter tracts. So the white matter tracts are tracts that connect the different regions of the brain, for example, Wernicke's and Broca's area. So for example, here in green are the tracts, the primary tracts that connect the regions associated with Alzheimer's disease. I guess you can't see my pointer. Can you see the pointer or no? So where you see the fornix, there's an arrow going lower right-hand side, and so that so that big thick green branch, as it goes in the inferior direction, it goes towards the hippocampus, which is a key part of in terms of Alzheimer's disease. So that thick branch is part of the cingulum, and the fornix is right there. So most of it, what you're seeing there, is the cingulum itself, and the cingulum goes all the way around. You can see in the posterior part of the brain, there's the word cingulum, and so the cingulum goes all the way from the hippocampus here to the posterior cingulate region, and then it arcs all the way to the medial frontal regions. Now, in this diagram, the green region going into the frontal regions is not going all the way into the frontal lobes, just really only in the posterior part of the frontal lobes, but the cingulum usually goes into the anterior parts of the frontal lobe. And then the fornix, you have this very tight loop inside, and this fornix connects the hippocampus to the thalamus, and the thalamus is a key area in terms of connectivity to the cortex itself. Okay. There's, for example, frontotemporal dementia. The tracks highlighted in red are important in terms of frontotemporal dementia, and the uncinate fasciculus connects the frontal lobes to the anterior temporal lobes. So that track is key, it's one of the most affected tracks with frontotemporal dementia. There's also the inferior longitudinal fasciculus, as you see here in the inferior part of the brain, that's also associated with frontotemporal dementia. It is also, you could argue also it's associated with Alzheimer's disease. Then in blue are the tracts associated with primary progressive aphasia and corticobasal degeneration. These are neurodegenerative disorders associated mainly with, one of the key impairments is motor function. And here, that a lot of these tracts going upwards connect to the motor tracts as well as the parietal regions. So again, with motion, movement, these are key regions for that domain. Again, so you can see that the various regions that we know are associated with disease are connected through white matter tracts in the brain. Next slide, please. Yeah. So that complicated figure you saw in the previous diagram can be simplified in a matter of speaking. It may not look simplified here, but if you look at let's look at panel B first. You can see that there are a lot of dots that are spread all over the brain. So the size of the dot, that means the diameter of the dot, indicates the strength of the connectivity, or how connected that region is to the rest of the brain, measured with white matter tracts. So they looked at one region and investigated how strongly connected, how many white matter tracts are connected to the rest of the brain. And what you can see here is it seems mainly posterior regions are very highly connected as measured with the white matter tracts. Now you can take all that information and convert it into the diagram that you see here in panel A, and you can define hubs as the center or localized center of networks or subnetworks. So for example, here we have 6 hubs. That is, within each circle, you see a center. And so for example, in circle number 3, the center is the left parietal superior temporal cortex. So that's the hub. That's one hub of this subnetwork. This particular hub, or this region, is very highly connected to neighboring regions, and this hub is also connected to the other hubs. Okay, so hubs are defined by how strongly connected they are to adjacent regions, and the regions that are most highly connected are the hubs. And here they've taken the 6 most highly connected regions and defined that as the key hubs in the brain. So if you look at circle number 2, you can see that the hub is the posterior cingulate. Okay. And on circle number 4, you have the center of the hub is the right posterior superior temporal cortex. So again, these are so you have a map of the brain in terms of how the regions are connected. So you can see, for example, that the hub is in the center of one of the circles, and you have other regions that are very highly connected to that hub, but are not necessarily connected to other regions that are located within the other circles, so to speak. Okay. So this is looking at the network in the brain using white matter tracts. If we go to the next slide, please. We can use the resting state networks. So that this is looking at the baseline activity within the brain and looking at the coherent brain activity. So anytime, you know, we have, you know, the brain per se is never at rest, but the different regions within a network are always are always interacting with each other, and so there's a coherent activity, the low frequency coherent activity that we can measure with fMRI. So this activity is intrinsic to the network, and it's large amplitude, very low frequency activity, and one can measure again with fMRI. It can also be measured with PET, okay, in different ways using a different technique. So to differentiate or define the networks, we look at the linear correlation among the different networks, and the regions that are have very high linear correlation are defined as belonging to one specific network. So if you look at the next slide, please. Again, these patterns of functional connectivity, again, reflect the activation or the baseline activation within each network. Now, what happens if there's a lesion due to, let's say, Alzheimer's disease or you know, frontotemporal dementia? The lesion will impair this functional connectivity or interaction among the regions. And this impairment in the network activity can be measured with resting state fMRI. So the big advantage of measuring resting state or the network activity in the brain is that a patient that's cognitively impaired is more likely to be able to lie quietly in the scanner compared to the case where you ask a person to do a cognitive task while they're in the scanner. It's much more challenging for a cognitively impaired person to do something within the scanner itself compared to just lying quietly within the scanner. So these approaches are very powerful and relatively easy to do experimentally. The real challenge comes actually within the analysis domain. If we go to the next slide, please. So these three panels show the key regions or the hubs that are measured using fMRI. So if you look at panel C, you see in blue the hubs or the most highly connected regions within the brain. And you can see in panel C, the first brain image on the left-hand side, that's the left inferior parietal cortex. Then on the next slide, the next image, you can see the posterior cingulate and then the anterior cingulate cortex in the frontal lobe. This would be in the left hemisphere, and then the next brain image is in the right hemisphere. And then the fourth brain image in panel C, you have the right inferior parietal lobe. So these are key regions. They're called hubs in the brain measured with functional MRI or resting state. Now, if you look at panel B, you have the blue blobs are these hubs that you see in panel C, but they're placed within the connections within the network of the rest of the brain. Okay, so you can see that diameter of these, in this case, they put the only five hubs, is much larger than the rest of the other regions, indicating that, again, it's a much highly connected region. So again, illustrating you can reorganize the brain in terms of a network, and the network approach is how highly connected are they to each other. And so when you study disease, if you have a lesion, let's say in region C, you try to understand if region C is lesion there in panel B, what is the effect on the rest of the brain or the regions that are connected to it. So next slide, please. So this network that I talked about in the previous slide, illustrated in blue, is called the default mode network. And what you can see here on this slide in red are the areas of the brain that have very high amyloid deposition in Alzheimer's disease. And you will see that the red regions have high amyloid deposition, and they match quite well, or they overlap quite well with the location of the hubs. So there's an overlap between the default mode network, and this network is the regions or the hubs as shown in the previous slide. They overlap with the amyloid deposition. So this was one of the first studies showing the overlap between a network and a pathology in neurodegenerative disease. This approach has been extended to other neurodegenerative diseases as well as to other psychiatric disorders. Okay. So if we go to the next slide. Now for this slide and the next slide, I'm going to show different resting state networks. So one of the key questions is how do you define a network, or what do you consider to be a network? There's in these days usually anywhere about 12 to 15 networks are considered to be the large-scale networks that people in general consider to be relevant for cognition and for neurodegenerative disorders. Now, you can subdivide these individual networks that I'm going to illustrate here into subnetworks or components. So, it's again, when you look at the literature, you will find the definition of networks tends to vary a bit from one to the other. But in general, this would be what you're going to see here, as well as for the case studies. It's usually what you typically see in most studies. So, for example, in row B, you have a dorsal attentional network. So that would be these yellow blobs in the parietal lobe and then frontal lobes. So they're called a dorsal attentional network because we know these regions are key for attention. So a lot of other previous literature indicating, again, these regions key for attentional function in normal individuals. You have row C is the big blob is there in the posterior cingulate. And this is, again, a key region that's associated with the default mode network. And so in this case, what you can see is this posterior cingulate region right here is, so the question is it supposed to be so asymmetric? The question is yes, because there's one on the left hemisphere and there's one in the right hemisphere. So we have here row number 2 is in the left hemisphere. I'm assuming the left side of the image is left hemisphere. And we should see one other network that's a mirror image, but in the right hemisphere. So we have the third row, and is part, we have this, what you would call the posterior cingulate cortex. So this could be considered to be one of the subnetworks of the default mode network. Okay. So we go to the next slide. A second. So the second, this would be the second row. You can see it's mainly activation in the motor cortices. If you go to the top row, that would be attention. So this is where things get a little bit complicated. This would be attention, but also it's actually including a bit of language. So Wernicke's area. So if you look at the top row, the third brain slice from the left-hand side, on the right-hand side, you can see there is this bigger red blob, and that would be Wernicke's area. So now, obviously, now you can see that actually the right-hand side of the image is left hemisphere. Okay, so my guess in the previous slide was wrong. So again, so this is an illustration that sometimes the definition of the networks can sometimes be a little bit messy. It depends on your data and the methods that you use for the analysis. If you look at the bottom row, well, the second from the bottom row, so that's early visual areas. So this would be visual function. And if you look at the bottom row, right there is again another visual network within the brain. Okay. So now if we go to the next slide. So is there an association between this cognitive function and resting state networks? So just the next, so the answer is yes. So there was a study a few years ago that analyzed on the order of about 15,000 subjects, and they mapped the different cognitive domains from these huge number of subjects, and they investigated if the networks related to the various cognitive domains match the resting state networks. Okay, so you can see here in this panel, so let's look, for example, at panel number 1 on the upper left-hand corner. The left column is the resting state network, and on the right-hand side column is the brain network using the cognitive domain data. Okay, so what you can see here is this visual network on the left-hand side matches one of the networks that they found using the brain activation data. So by brain activation data, I mean data where that was measured when people are doing a cognitive task. For example, looking at images, or doing a memory task or an attentional task, and so on and so forth. On average, there is this network activated. And you can see that there's a very nice correspondence between the two. Again, the second panel, you can see early visual regions. Again, one using the resting state data, the left-hand side column, the right-hand side column, the brain activation data. And you can see through the 10 panels that there is a very nice overlap between the two. Now, as we'll be using this data to solve to answer the 4 cases, I'm just going to mention a few that will be important. So panel number 4 is the default mode network. So you can see the posterior cingulate cortex, the anterior cingulate region, anterior frontal regions, and lateral parietal regions. So the left-hand side would be the resting state, and the excuse me, the right-hand side is the brain network, the left-hand side, the resting state network. Then we have, for example, panel 9 and panel 10. These are the dorsoattentional networks. One is for the left hemisphere, the other one is for the right hemisphere. Okay, so left-hand side of the image is right brain. Okay, so these are two dorsoattentional networks. Panel 6 is motor network. Panel 7, well, let's see, panel 7 would be, I think the closest we have here to a language network. Okay. So if we go to the next panel, please, next slide. You know, so the previous slide was correspondence between cognition and the different networks. Similar approach was done with the 5 different dementia groups. So AD, frontotemporal, semantic dementia, progressive non-fluent aphasia, and corticobasal syndrome. And so they looked at the structural atrophy. You know, is the structural atrophy in the brain, you know, associated with specific brain networks? And are there specific networks associated with different diseases? If we go to the next slide. So in this study, they showed that if you look at the top row, that's the data from the patient groups. And the middle row and the bottom row is the data from young, healthy individuals. So the middle row indicating the connectivity using resting state fMRI. The bottom row looking at connectivity using structural MRI. And you can see that the blue on the top row overlaps with specific networks as measured in the young, healthy individuals, both in the yellow and the green. And so the conclusion is that specific neurodegenerative disorders are, at least in the early stages of the disease, affect specific brain networks. And so that explains the association between specific pathologies at the molecular level and the associated cognitive and clinical symptoms that a patient may suffer from. So you have amyloid pathology and tau neurofibrillary tangles forming in AD. You know, there's the reason why that pathology is associated with memory impairments is that the pathology affects regions associated with a default mode network, which is key for memory function, among other things.